recording. All right. There we go. Let's move this here, this here. So what I'm going to do is I'm going to go through a little bit of a summary of the theory, and then we'll smash through a few questions. So to begin with, I'm going to start over here with a chloroplast. Let's do it in green, because this is good. There are a bunch of features here that I have. So I've got feature number 1, feature number 2, feature number 3, feature number 4, feature number 5, and feature number 6. You're going to chuck in the chat any number and tell me if you know what any of those things are. Okay, so number 1 is referring to one of those things. You're going to have to tell me the number, but I'm presuming you said this. I've got that one there. You can be really tricky and give me 6 or 5 or 4 or anything like that. So I've got my granum. Which one is the stroma? Oh, the stroma isn't labelled, is it? So I'm going to put a 7. Here's my stroma. Did I give you this at the beginning of the year? Can you thumbs up in these reactions anymore now? I did give you this earlier. No, you don't think so? Normally I would print this, I think. I drew it years ago and it's helpful as a little bit of a summary sheet for us. So I've got a ribosome here. If I want to be really precise, I'm going to call it a 70S ribosome. 5 is the outer membrane. 6 is the inner membrane. And number 4 is the only one I'm missing. What does it kind of look like if I give it some more lines? It is a nucleic acid. It's the circular DNA chromosome. Okay, so I tend to start off revising photosynthesis by focusing on the chloroplast and what's happening within it. So the next part of the question that comes along after describing my structure is to, I'm just going to move this up a little bit, is to give us some evidence for bacterial origins. So we think that the chloroplast was once a cyanobacterium. So it was once a cyanobacterium. Yeah, and that means that they had the ability to photosynthesize. So whatever was present in these was the ability to photosynthesize. So what is some of the evidence in here that indicates this might be an old bacterium? Double membrane, thank you. I'm looking for at least 2 more things. The 70S ribosomes. So you can call them 70S ribosomes, or you can just say ribosomes that were the same in bacteria. The double membrane indicates engulfment. And the circular DNA, or the circular chromosome, circular DNA chromosome is another great piece of evidence. Now, the good thing is all of these things are also present in a mitochondria. So once you remember them for the chloroplast, although this inside of a mitochondria looks different to a chloroplast, they both have the same evidence for their bacterial origins. They both have double membranes, they both have these 70S ribosomes, or these ribosomes that are the same as bacterial ones, and they both have circular DNA chromosomes. Okay, now embedded in those thylakoids, those stacks, number 1, so a thylakoid is one of those discs, and a granum is the word, so number 2 is the word for a stack of discs. And embedded in them there is a pigment. The pigment in a green plant is embedded with chlorophyll. Okay, so in these thylakoid membranes, they are chlorophyll, and they are the things that actually give the green color. So chlorophyll absorbs the red and the blue wavelengths of light, and it reflects the green, and that's why we see plants as being green. Are you just going, oh yes, now I kind of, oh, there's something about reflecting light that you talked about like 6 months ago that I may have remembered. Now, we did photosynthesis around the time that you guys got launched into this mode the first time. All right, does anyone have any questions about structure or evidence? Okay. The next thing is my overall equation. So I'm going to have A, B, C, and D, and then I'm going to have E and F for you to give me some answers. So you tell me what you think is A, B, C, D, E, or F. But C is glucose. Ooh, that's annoying. You've got oxygen as D. You've got carbon dioxide as B. And water as A. So I just need some stuff for E and F. E is light energy, and what would F be? Chlorophyll. Good. So, look, the overall equation has come up once or twice, but it's generally not a big deal with photosynthesis. We don't think too much about the overall equation. What is more important is this glorious little table that I've got for you here. Because this overall equation, the reason I like to go through the overall equation is because it can help us to fill in this table. So we've got to think the inputs are the things that are on the left of the equation and the outputs are the things that are on the right of the equation. So I've filled in part of it for you. There are only two stages in photosynthesis. We've got the light dependent stage and the light independent stage. So the light dependent stage, this is going to happen in that thylakoid membrane. So over here, this is where it's going to happen, on that thylakoid membrane. It's nice and I managed to paint it green. How good am I? So on the thylakoid membrane, why is it happening in the thylakoid membrane? Because that's where all the chlorophyll is. Okay, and we know that we need chlorophyll and it's light dependent. So this is also where the light goes in. So I think, I'm trying to think of a good way to help us remember this. We'll see how we go. So the water splits in the presence of light. Yeah. So that process, think about what is water made of? It's made of H2 and O. So we know that we're not going to get any H's out because our two outputs that we can choose from are glucose and oxygen. So the water is actually going to become oxygen. And where did the H's go? The H's went onto the NADPH here. In addition to that, I also loaded some more coenzymes, ATP as well. Okay, does that kind of ring bells? And what I'm going to suggest to you at the end of this is this table in particular, and the one for cellular respiration, this is what I would draw out and stick inside your shower. Yeah, because it's just practice of getting to what are the inputs and the outputs of each particular reaction. So the oxygen is actually a bit of a waste product. What we really want from this light-dependent reaction is the NADPH and the ATP. That was the whole point of this. And what they do is they actually fire up the light-independent reaction or the Calvin cycle. So now we move out into the stroma of the chloroplast, which is this pink region out here. Okay, there's all this pink region. Pink, 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 pink. I'm just going to have fun colouring this in. Tell me if I go too fast, girls. So when those NADPH and ATP go in, they need to use their energy to do something. So what was the other ingredient, the other input, the other thing that was on the left-hand side of my equation? CO2. So carbon dioxide goes in here. Now these loaded coenzymes that have come with all their energy, they're going to use their energy to turn that carbon dioxide and also the H's from the NADPH, they're going to turn that into glucose. Okay, so this is the whole point of photosynthesis, and I'm going to use another color now. So this is where there's a dot point that asks you, what is the point of photosynthesis, or what is the purpose? This is the purpose right here, to make glucose. Now, when they drop off their hydrogen ions, if you're NADPH, if you drop that off, you're going to become NADP plus again. Yeah, so there are two coenzymes, and I might just write a little bit about this somewhere else in a second, involved in this. And the point of them is, why do we need them? So so these are my coenzymes. NADPH can be put into NADP plus. These are the loaded forms, loaded. These are the unloaded forms. And this one is ATP is the loaded form, ADP plus PI. So what is the purpose of each one of these? So the purpose of the first one of these, NADPH going to PI, the purpose of this is to transfer H plus and energy, sorry guys, from light dependent to light independent reaction. That's its purpose. That's right, purpose there. So the next question is, what's the purpose of ADP and ATP? They don't transfer hydrogen ions, but their purpose is to transfer energy from LDR to LIR. So that coenzymes dot point is actually part of the previous section. So it's a part of the enzyme section, but it's really hard to teach it there. So I often teach it up here. Does anyone have any questions about this table in particular? I have yet to come up with an excellent mnemonic that helps you remember these things. pause the. Very chatty now. I'm taking silence to mean we're all good. So I hate to say it, girls, that's the hard bit for photosynthesis. That's it. I've just, essentially, if I was at school, I would have printed out this blank sheet for you, and it would have taken up one A4 piece of paper, maybe an A3 if I felt like being generous, but you could fit this on half an A4 piece of paper. And the thing is, once you get this data, this information in your head, then when you start to do questions, it becomes a lot easier. So the only other part of this, it's just there. The only other part of this section of the course is the factors that affect photosynthesis. And so what we get is, oh, sorry, wrong way. We always measure our light intensity, or the thing that we're changing, our independent variable is always going to be on the x-axis. Yeah, so light intensity will go on my x-axis. The other factor that can affect, one of the other factors that can affect photosynthesis is temperature, and the final one is CO2 concentration or CO2 availability. Yeah, so if it's something I'm changing, I'm going to put it down the bottom. And so what we get with light intensity is we get this relationship. So what goes on the y-axis is ROP, which stands for rate of photosynthesis. Yeah, and as the light intensity increases, I'm going to do it this way. As the light intensity increases, the rate of photosynthesis will increase. Yeah, but does the graph just go as a straight line, or does something else happen? I'm hoping that you're recalling that the line actually does something like this. It plateaus. And what that means is that up at this stage here, another factor is limiting the rate of photosynthesis. In this pink section, in this section here, light intensity is what's limiting the rate of photosynthesis. Yeah, we get that. Plateau is like one of those really weird words to spell. Just going to pause, and that'll make it a bit better. All right. So I'm going to skip temperature, and I'm going to go to CO2 concentration. And what happens with this graph is, again, rate of photosynthesis is on the y-axis. But this time, as CO2 concentration increases, because it's an input, see how it's an input here on the left? As CO2 concentration increases, the rate of photosynthesis increases. So it's the same as that beginning bit of the, yeah. So CO2 concentration is the limiting factor here, is limiting the rate of photosynthesis. But it does the same thing. It does this plateau. And the reason it does this plateau, even though you keep increasing your carbon dioxide concentration, so you just keep giving it more and more carbon dioxide concentration, it's because another factor is limiting the rate of photosynthesis. And this could be, e.g., light or temperature. With this one, back here, it could be, e.g., CO2 concentration or temperature. So can you see how the light intensity graph and the carbon dioxide graph are exactly the same? All you have to do is look at what's on your x-axis, and that'll help you with those. Now, the temperature graph looks a little bit different. The temperature graph goes up. So as temperature increases, the rate of photosynthesis increases. Then it gets to a top bit, and then it goes bang, and goes down again. So we get that same pink bit where, as we increase temp, we increase the rate of photosynthesis. Oh, why do I always go rate? So frustrating. Oh, so annoying. Have you ever seen Dr. Forward? She's got like the best handwriting on an iPad. Let me just pause this one, and then I'll start recording for the next one. All right, so what happens is we get to the top here, and this yellow section here. What is different about this is it is the optimum temp. And then this graph has a third section to it. It has this blue section. And here we increase the temp and it decreases the rate of photosynthesis. Why is it going to do that? Because the enzymes, there are enzymes involved in this process, and they are going to denature. So essentially, there's only one new graph that you learn for photosynthesis compared to enzymes, and that's the graph for the limiting factors of light intensity and carbon dioxide concentration.